Yeah, our study at THJ was to try to understand or get novel insights into the origin of CLL and how CLL evolved by studying CLL patients in which we do not identify a single CLL clone, but multiple independent clones. So it's patients that don't have a single B-cell neoplasm that have multiple CLL at the same time, but of independent origin. Because this was already suggested that a fraction of patients had this phenotype or this manifestation of the disease. We wanted to understand which was the magnitude of this phenomenon and which were the molecular features of these different clones. Yeah, in our study, we first started analyzing more than 350 patients with CLL or what's called monoclonal B-cell lymphocytosis, which is the preceding phase to CLL. And also we compared what we found in the patients against a cohort of more than 120 healthy individuals, age-matched healthy individuals. And we first studied these samples from these patients and healthy individuals using next-generation sequence of the monoglobulin gene, which basically allows us to understand or characterize the architecture of the CLL. So to identify which patients have one CLL, which have multiple clones with different immunoglobulin gene. And when we characterize this, we found that in CLL, for instance, there is around 12% of patients that have multiple CLL clones. Then the next question was to try to understand which was the molecular features of these different clones. And for that, we performed different single-cell technologies. We performed single-cell RNA-seq, single-cell DNA sequencing, and single-cell whole genome sequencing to understand which was the transcriptome and driver alterations of these different clones. Yeah, the results that we found was that beyond this 12% of CLL patients that present this oligoclonal pattern, so multiple independent clones in their peripheral blood, when we went deep below clones that were very, very minor, we also identified that in patients with CLL, there is a bias in the B-cell repertoire in the patients compared to the healthy individuals. So it's not only that some patients might have more than one expanded clone, but that let's say, to make it general and easy to understand, that most of these B cells in the peripheral blood are biased towards CLL characteristics at an immunogenetic level. And then when we perform all these single cell technologies, we found that these clones or these B cell clones that are different from the main CLL population also have the transcriptomic profile of CLL based on the single cell RNA sequencing. We also observe that these different clones, even if they are very small in size, they can carry driver alterations of CLL that we typically observe in patients with CLL. And some of these driver alterations might be associated to aggressive disease. For instance, TP53 mutations or the LGLB321R110 mutation can be found in very, very small clones. And finally, what we found also using single-cell whole genome sequencing is that these different clones do not share any single somatic mutation in their genome, so suggesting that they originate from very independent or completely independent cell of origins. And using this technology also allows us to understand when these different clones start to emerge during the lifespan of the patient. And we find, for instance, that in some cases, the initial driver alterations in some of these clones were acquired before the age of 30 in patients that were diagnosed at the age of 60. And we could observe also how some of these clones start to grow and expand decades before the diagnosis of the disease. And just to give you some numbers, for instance, we found that in some patients with CLL, we identify up to 9 different CLL clones. So different clones with different monoglobulin genes, all of them with the phenotype of CLL. Yeah, we started to analyze this phenomenon because we think that, well, there is some evidence that in the early phases of CLL, there is much higher oligoclonality. That is what we also found, that in the MBL, there is like 40% of patients presenting this oligoclonal pattern. So by understanding this multiple oligoclonality found in CLL, it's a way to understand the initial steps of CLL development. So going back and let's say in time to understand the origin of this disease. And doing so, we can try to understand which are the initial driver alterations that are found in these clones, what makes some of these clones expand faster than others. So let's say understand the initial steps of CLL origin and how these clones evolve. And this could also allow us in the future, if we have good drugs that could target this CLL early, and this is something that maybe in the future is shown. All this technology that we are developing here, the single cell technologies that allows us to identify these minor clones and so on, would allow us, for instance, to think in the future in potential early detection of CLL or early detection of more aggressive clones, and this could lead to potential the development of potential early intervention schemes.